Tissue engineering is the study of the growth of new connective tissues or organs from cells and a collagenous scaffold to produce a fully functional organ for implantation back into the donor host. This technique will allow organs to be grown from implantation rather than transplantation and hence free from immunological rejection. The starting point for any tissue-engineered organ is the harvesting of small amount of tissue from the future recipient of the tissue-engineered organ. This could be as small as 2 mm punch biopsy for some applications. Cells from biopsy are then cultured from explants or a collagenase digestion to create a cell bank. These cells are then further cultured on a collagenous substrate under the correct physiological condition to form tissue-engineered construct for implantation. The process is carried out in a tissue culture facility to maintain a sterile environment. Cellular biochemical and physical activity can be enhanced by the addition of growth factors or cytokines, also by the use of physical stimulation.
What is tissue engineering? Tissue engineering is the use of a combination of cells, engineering and materials methods, and suitable biochemical and physicochemical factors to improve or replace biological functions. While it was once categorized as a subfield of biomaterials, having grown in scope and importance, it can be considered as a field in its own right. While most definitions of tissue engineering cover a broad range of applications, in practice the term is closely associated with applications that repair or replace portions of or whole tissues, bone, cartilage, blood vessels, bladder, skin, muscle, etc. Often, the tissues involved require certain mechanical and structural properties for proper functioning. The term has also been applied to efforts to perform specific biochemical functions using cells within an artificially created support system, an artificial pancreas, or a bioartificial liver. The term regenerative medicine is often used synonymously with tissue engineering, although those involved in regenerative medicine place more emphasis on the use of stem cells or progenitor cells to produce tissues. Goals of tissue engineering. Tissue engineering evolved from the field of biomaterials development and refers to the practice of combining scaffolds, cells, and biologically active molecules into functional tissues. The goal of tissue engineering is to assemble functional constructs that restore, maintain, or improve damaged tissues or whole organs. Artificial skin and cartilage are examples of engineered tissues that have been approved by the FDA. However, currently they have limited use in human patients. Regenerative medicine is a broad field that includes tissue engineering but also incorporates research on self-healing, where the body uses its own systems, sometimes with help foreign biological material, to recreate cells and rebuild tissues and organs. The terms tissue engineering and regenerative medicine have become largely interchangeable, as the field hopes to focus on cures instead of treatments for complex, often chronic, diseases. This field continues to evolve. In addition to medical applications, non-therapeutic applications include using tissues as biosensors to detect biological or chemical threat agents, and tissue chips that can be used to test the toxicity of an experimental medication. Importance of tissue engineering. Tissue engineering is a radically new concept for the treatment of disease and injury. It involves the use of the technologies of molecular and cell biology, combined with those of advanced materials science and processing, in order to produce tissue regeneration in situations where evolution has determined that adult humans no longer have innate powers of regeneration. Tissue engineering, however, along with some other aspects of regenerative medicine such as gene therapy, has yet to deliver real successes in spite of a considerable science base and investment in the commercial infrastructure. This article addresses the underlying issues of benefit and risk in tissue engineering in an attempt to understand why this situation has developed. There is an inherent, virtuous logic to tissue engineering that sounds too good to be true. By my definition, tissue engineering is the persuasion of the body to heal itself, achieved by the delivery to the appropriate site of cells, biomolecules, and supporting structures. It specifically involves the regeneration of new tissue to replace that which has become diseased or injured, the significance of which is that we, as adult humans, do not normally possess this ability. We may repair ourselves under some very limited circumstances. For example, bone fractures and injured skin may undergo repair, but, even when this does occur, this usually involves non-specific reparative tissue, scar tissue, rather than the regeneration of the specific functional tissue that has been affected. The essence of tissue engineering is that those cells capable of initiating and sustaining the regeneration process are switched on, perhaps through growth factors or genes, so that they generate new functional tissue of the required variety. This may be achieved with the help of a scaffold or matrix to guide the geometrical or architectural shape of the new tissue, and may take place on a customized basis at the site of the injury in an individual patient, or on a more industrial scale in an ex vivo bioreactor, where the resulting tissue construct is reimplanted into the patient. Issue in tissue engineering. Tissue and organ failure, produced as a result of injury or other type of damage, is a major health problem, accounting for about half of the total annual expenditure in healthcare in the U.S. Treatment options include transplantation, human or xenotransplantation, surgical repair, artificial prostheses, mechanical devices, and in a few cases, drug therapy. Ultimately, however, major damage to a tissue or organ can neither be repaired nor long-term recovery effected in a truly satisfactory way by these methods. Tissue engineering is emerging as a significant potential alternative or complementary solution, whereby tissue and organ failure is addressed by implanting natural, synthetic, or semi-synthetic tissue and organ mimics that are fully functional from the start, or that grow into the required functionality. Initial efforts have focused on skin equivalents for treating burns, but an increasing number of tissue types are now being engineered, as well as biomaterials and scaffolds used as delivery systems. A variety of approaches are used to coax differentiated or undifferentiated cells, such as stem cells, into the desired cell type. Notable results include tissue-engineered bone, blood vessels, liver, muscle, and even nerve conduits. As a result of the medical and market potential, there is significant academic and corporate interest in this technology. Advantage and disadvantage of tissue engineering. The pros of tissue engineering are obvious. Engineering tissues can potentially help a person conquer a disease or illness. Tissue engineering may even be able to cure extreme and mild arthritis in patients that receive this sort of treatment. Tissue engineering also has the capability of prolonging our lives and making the general quality of our lives much better. The cons of tissue engineering are fewer than the pros, but they are present. Two main cons to tissue engineering are often discussed. One is the possible presence of latent or hidden diseases or illnesses in the base tissue. There is the possibility that if foreign body tissue was used to reconstruct particular tissue matter for a patient, there could be hidden disease or other hidden illness within that tissue matter. This is something that scientists are working hard to prevent. However, latent diseases are troublesome to find with the technology.